Hello everyone, let us answer this photosynthesis question from a previous life sciences grade 11 question paper. 3.3. The diagram below shows a variegated leaf being tested for photosynthesis. Now, a variegated leaf is basically a leaf that has a different color on part of the leaf than on another. So what we're basically having here is a leaf that is containing two colors, green and white. So the outer part of the leaf is white, meaning it does not contain chlorophyll. Then the inner part of the leaves, this gray shaded area, is actually green, meaning it contains chlorophyll. Then the chlorophyll part of the plant is also covered with a black paper, as we can see on our diagram. So that's basically what we're having. So I'm not sure if I'm going to win, but I'm going to try and draw so that you can see exactly what is taking place in this experiment. So that's the leaf. Now the outer part of the leaf, let me do it like this. So the outer part of the leaf, this part is white and this part has no chlorophyll. There is no green pigment. Then the inner part of the leaf, I'm going to try and use a green color. So this inner part of the leaf is actually green. Remember, in life sciences, we do not color in our diagrams. I'm just doing this to show you and to explain this particular investigation. Okay, there. Sure you can see what's happening. So this particular part of the plant is green, and this one meaning it contains chlorophyll. It contains chlorophyll. Remember, chlorophyll is that pigment that will assist with absorbing energy from the sun. So what you also then have in this particular investigation, the green part of the leaf is covered with black paper. So this green part of the leaf is covered with black paper. So if you place a black paper, so it's going to cover this green part of the leaf. So obviously when you're covering the leaf, you need to make sure that you cover it on both sides. So the dorsal side and the ventral side, the front side and the back side of the leaf. All right. Yes, you see, now at least it's clear. All right. So let's see. Before the experiment, the leaf was destarched. Okay. So the first question is saying we must give one reason for destarching the leaf. So why do we need to destarch the leaf before we begin with our experiment? So obviously that is obviously to remove starch, so that the starch present afterwards. Remember, we're going to destarch the plant, conduct an investigation, then test for starch. So that's how we do the whole experiment. So we obviously destarching the leaf to remove starch so that the starch present afterwards, shows that photosynthesis has occurred. Or you can even say to basically make sure that the starch tested at the end of the investigation is the only one that was produced during the investigation. So we allow the plant to use up all of the starch, convert it into a glucose and use it up, so that by the time we begin with our experiment, the plant contains no starch. So we are starting all over. We are going to expose the plant to the sunlight and allow it to photosynthesize. So we do not want the starch that was previously formed to disturb our investigation. We can even say that the reason we are destarching the plant is to make sure that the leaf is free from any starch that was previously made, or the leaf must be free of starch at the start of the experiment, so that evidence of the need for the factor being investigated can be provided during the experiment. So I think the easy answer, guys, is to remove starch. That's it. Is for the plant to use up all the glucose to ensure that there is no starch present at the beginning of the investigation. I think that's the easiest answer, and you will still get a mark for it. Then B, describe how the destarching is done. How do we destarch a plant? Okay, so we then take a variegated plant. You need to mention that it's the variegated one because we need a plant that contains leaves with two colors, the green color part and the white color part. So we basically take the variegated plant, the variegated plant, and we place it in a dark cupboard for 48 hours. The variegated plant will be placed in a dark cupboard for 48 hours. You need to include the hours, guys, 48 hours. So we basically isolate this plant from photosynthesizing, meaning it will use up all of the stored glucose, which is obviously stored as starch in that 48 hours. Then 332, state the factor being investigated. State the factor that is being investigated with reference to the region of the leaf marked B. Marked B is the region that is covered with black paper. So that means we are basically investigating light. We are investigating light or the need or the necessity of light during photosynthesis. So you can say the need of light during photosynthesis. Okay, because how this investigation is conducted, the part that is covered with the black paper obviously won't be able to receive energy from the sun. And the part that is not covered with the black paper, the green part, contains chlorophyll, and radiant energy will be absorbed and photosynthesis will take place. Right? That's what we are looking at. The white part, guys, it will be almost the same as the black part because the white part contains no chlorophyll. That means no photosynthesis will take place there. Okay, so describe one precaution when covering the leaf with black paper. So I think here you just need to make sure that the black paper is placed on both sides of the leaves. So you place the black paper, or ensure that the black paper is covering both sides of the leaf. So the black paper must cover both sides of the leaf. For 2 marks, suggest the possible results or result of the experiment in region B. Remember the results of the experiment, guys, it's when we test for starch and they are asking about region B. Region B is this area of the leaf that was covered with black paper. So how we then test for results? We actually test for starch, and the step 1 is from this particular plant, we are going to take one variegated, actually it's this leaf that was covered because chances are the other leaves were not covered with black paper. So this leaf, you will then remove it from the plant, boil it in water, then boil it in alcohol, then rinse it in cold water. Yeah, it's cold water. Rinse it in cold water, then add drops of iodine. Now, you're basically telling us about the results of the experiment. When you add drops of iodine, what is happening there? Is there a change of color? So that's exactly what you need to indicate to us. So since this part was covered with paper, that means photosynthesis did not take place. So when we add drops of iodine, we are expecting the iodine color to remain reddish brown. And when it remains reddish brown, it indicates to us that there is no starch present. When there is no starch present, that tells us that photosynthesis did not take place. So if we take it back to the factor that we are investigating, that means in this region of of the leaf, photosynthesis did not take place. And the reason for that is because this region of the leaf did not receive light. And if we're to add drops of iodine on this region of the plant or of the leaf, we would expect the iodine color to change to a blue-black color. So the color change on this area will basically be the iodine remaining reddish-brown. So the iodine remaining reddish brown is indicating to us that there is no starch present. And if there is no starch present, that means photosynthesis did not take place. No photosynthesis, right? And that is proving to us that light is needed for photosynthesis to take place, because this part did not receive light and photosynthesis did not take place. Now, region A, which is green. So if we're to add drops of iodine there, the color change will be blue-black. And if iodine changes to a blue-black color, that tells us that starch is present. And if starch is present, that means that photosynthesis took place. Photosynthesis took place. Okay, I hope that is clear. So your answer here would be the parts covered in black, in a black paper rather, will turn reddish brown, or the iodine will remain reddish brown,